Alright, hello. My name is Dhanya, and I'm going to be presenting on cell communication in pancreatic cancer and the basics of cell signaling as well. So let's cover the basics of cell signaling first before we get into how it applies to pancreatic cancer. So there are two main types of cell signaling, which are local and long-distance signaling. So forms of local signaling include direct contact and local regulators, and they usually work in tandem. For animal cells, direct contact local signaling would be through gap junctions, where signaling molecules and ligands can move between the cytoplasm easily. And for plant cells, it would be through plasmodesmata, which is the same concept. There is intercytoplasmic substance transfer in both. In terms of local regulators, these are the signaling molecules or growth factors that actually travel in between the cells. So they are short distance and they can be used in synaptic signaling and paracrine signaling. For long distance signaling, they're usually used in the endocrine systems of organisms. So they would travel through the circulatory system for animals and for plants through gas or the vascular tissue. And one example of long distance signaling in terms of endocrine systems would be for hormones. So here we can see a diagram of how long distance signaling would work. So the endocrine cell would release the signaling molecules, which then travels through the bloodstream, and it goes to the appropriate cells. One common misconception is that these signals only travel to specific cells. However, the signaling molecules are available to all cells, but only certain cells have the receptors on the plasma membrane in order to process that signal, transduce it, and then create a response, which are the three steps of cell signaling, which we will look at now. So as I said before, the three main stages are reception, transduction, and response. So different forms of reception in cells can be through G protein coupled receptors, ligand-gated ion channels, and intracellular receptors. And intracellular receptors are usually used when the ligand or signaling molecule is hydrophobic, so it can easily pass through the membrane, and therefore it doesn't need a protein receptor, transmembrane protein receptor in the membrane itself. So how G protein coupled receptors work is important for pancreatic cancer because that is actually one of the things that goes wrong in terms of cell signaling there. So a signaling molecule will bind to the transmembrane protein receptor, and a GTP molecule, which is similar to ATP in that it carries a phosphate group as its potential energy source, will phosphorylate a G protein that is binded to this receptor. And then this inactive G protein is now active. It binds to an enzyme nearby, and it then begins the transduction stage of cell signaling. So what transduction means is that it's basically multi-step pathways to transfer or relay the signal. And one thing that you should not mistake is that transferring the signal does not mean transferring a physical molecule or some sort of signal. It rather means that there is a series of shape changes in proteins that eventually leads to a transcription factor being formed in the response stage of cell signaling. So as we can see in this diagram over here, there are the reception, which is with the signaling molecules and GPCR or ligand-gated ion channels, etc. It then gets transduced by changing the shapes of proteins based on each protein interacting with another protein. And then that quote-unquote signal can be responded to in the nucleus or the cytoplasm. So in the case of pancreatic cancer, it is responded to in the nucleus. So in the nucleus, usually transcription factors are how responses are made because they create new proteins to be coded, and that creates an overall response because proteins will affect like the overall response of the cell. Okay, so now let's get into cell signaling in terms of pancreatic cancer itself. So before we go into what exactly happens, let's go over what pancreatic cancer would look like or present itself like in a patient. So Usually, pancreatic cancer is unfortunately caught only in late stages, such as stage 4, and this actually causes it to be the number 3 leading cause of cancer-related deaths in the United States alone. So some of the symptoms, if it is caught, are jaundice, which is the yellowing of skin or the eyes, the whites of the eyes, due to excess bilirubin, which is a protein that is created by the pancreas and the liver, which shows excess bilirubin would show damage to those organs. Another symptom would be blood clots in the limbs or extremities, onset of type 2 diabetes, or worsening of existing diabetic symptoms. And then pale stools show that there's bile leakage into stools from the liver and pancreas, which obviously shows that there is some kind of ductal tumor or damage to the liver or pancreas. And then dry skin is obviously not as obvious a symptom for you would think cancer, but it is one of the symptoms. All right, so let's go into where pancreatic cancer gets its genetic origins. So gene mutations for pancreatic cancer are autosomal dominant, which means that they are not in the sex chromosomes, but they are dominant in terms of traits. So there's a large variety of research on which gene mutation contributes to the eventual development of a pancreatic tumor, but one of the widely agreed upon ones would be the KRAS gene, which is thought to contribute to early tumorigenesis or early formation of the tumor. And this gene is on chromosome 12p, which you can see over here. So what this gene does is it codes for a GTPase protein, which, as we saw before in the cell signaling stages, is related to the GPCR cell signaling, and it causes a malfunction in that signaling process. So what happens is that growth factor stimulation from the external fluid causes this membrane protein to be activated, and therefore the signal transduction through the GPCR causes eventual cell cycle progression, extracellular matrix changes, and it stops cells from committing apoptosis when they need to, those are 3 obvious signs of unhealthy cell growth and would obviously lead to a chain, an entire chain of cancerous growth, because it affects the cell cycle and the extracellular matrix, which would affect cell signaling, etc. And what's interesting is that this is just one gene that has been identified as causing the early tumor formation in pancreatic cancer. And there are other ones, but this is just one of the widely accepted ones. So let's go a bit into what happens. So basically the KRAS gene-coded protein, it receives growth factors from outside, and then it is triggered and it starts transducing and causes downstream effectors which amplify the signal. And then transcription factors are made in the nucleus, which then in turn produce more growth factors and chemokines. And what happens is basically a positive feedback loop where these chemokines and growth factors feed back into the cell as more ligands, and then that interacts again with the nucleus, creates more transcription factors, and therefore you can see how the KRAS gene eventually affects cell cycle progression and cell signaling and all the things that lead it to be a cause of pancreatic cancer. So in terms of risk factors and treatments for this type of cancer, risk factors would include smoking, diabetes, usually type 2 diabetes, obesity, pancreatitis, and ethnicity, specifically the African American ethnicity, is usually more at risk for this cancer. So what's interesting is that type 2 diabetes, sometimes type 1, and obesity lead to pancreatitis, which is the inflammation of the pancreas, which would obviously affect how cells signal with each other, and any type of damage to cells is usually bad. Like in skin cancer, for example, those types of damages can obviously lead to abnormal cell signaling and, unfortunately, cancer. So some of the treatments that are used today to slow this type of cancer are surgery, which is pancreatomy, or removal of part or all of the pancreas, chemotherapy and radiation, which is standard for all types of cancer. Unfortunately, it does not target only specific cells and can be harmful to healthy cells as well. And the last form would be targeted therapy, which is better than chemotherapy and radiation in a sense because it acts against specific cells and not all presently dividing cells like chemotherapy does. So, what targeted therapy does is it focuses on blocking certain cell signaling pathways, such as the KRAS gene's GPCR pathway. And this is what makes cell signaling so important to diseases like pancreatic cancer and all types of diseases, because actually 60% of all modern medicines used today use GPCR pathway intervention as the medicine. So that's what makes cell signaling so important, and I hope you learned a bit about pancreatic cancer and cell signaling today. Thank you.